So this video is for the Aseptic technique lab. Just going to do a couple demonstrations here. Firstly, when you get it in, you would sanitize your workstation. So this is my spray bottle full of sanitizer, and you'd have everything off the bench, spray it down, wipe it thoroughly, make sure it's clean, sterile. And then you would grab your supplies. So what we have is our inoculating loop and the test tubes and test tube rack. So let's go here. And then also the micro incinerator, which is this guy. So firstly, you take your inoculating loop, hold it like a pen. You place it inside the micro incinerator for 10 seconds to sterilize it. Let it cool for a minute so that you don't kill the bacteria you're trying to transfer. You then take your stock culture out of the test tube rack and remove the lid with your pinky, like this, like that. Sterilize the top of it for a few seconds in front of the micro incinerator. Take your inoculating loop, dip it inside, make sure you get the culture on there. Once you have the culture on the loop, you take the tube again, sterilize it before you put the cap back on. Place the cap, goes back in the test tube rack. You take your media that you're transferring to, same thing, take the lid off with your pinky, sterilize the top of it for a few seconds, take the loop, place it inside, and kind of spin it to make sure that you get all of your culture into the media. Remove the loop, sterilize the top of it for a few seconds, and replace the cap. Goes back in the test tube rack. And lastly, you would sterilize the inoculating loop, just like you did before, for 10 seconds, and then let it cool down. The second thing is transferring from a culture inside a test tube to an auger plate. So similar process. Of course, first you sterilize your loop. Once that's done, let it cool for a minute. Take your culture, remove the lid, sterilize the top of it for a few seconds. Take your loop, get your culture on there, remove it, sterilize again for a few seconds, replace the cap. Then you take your petri dish that you're transferring to, and you carefully lift up the lid like a clamshell, not too much, you don't get anything in there. Take your inoculating loop and just scrape it across the surface until you have a visible indent where you know your culture is. Remove this. Put the petri dish down and sterilize one more time for 10 seconds, and then let it cool for a minute. Lastly, transfer from one dish to the other. So similar process here. Take the loop, put it inside the micro incinerator, remove it, let it cool. Take your first dish that has your culture in it, open the lid up, and get just across the surface. You don't need a ton of it. Make sure that you have it on there. Place the lid. Take your media that you're transferring to, open that one, and same thing, just scrape it right across the surface until you see a just barely visible indent across the surface. And then you close this, and when you put it inside the incubator, you would keep it lid side down so that no condensation builds up. And of course label it and all that stuff. And then finally sterilize this one more time for 10 seconds. Remove it, let it cool, and then once you're done transferring, you'd remove everything from the bench like before and sterilize it one more time. So take your spray bottle, spray, wipe it all down, and you're good to go.